So welcome everyone. This is supposed to be session 5.3 breakout. I'm talking about the morphological basis of lung adenocarcinoma subtypes. So a session outline, we're going to talk a little bit about a paper that was published about 2 months ago called the Morphological Basis of Lung Adenocarcinoma Subtypes. And then we're going to explore the model that was used in that paper. I put this slide deck, as well as a zip file with the Computational model up in the student material folders for today. Feel free to go ahead and probably might want to grab the model folder, but not necessarily the PowerPoint. So you can grab that as well. I'll have to update the PowerPoint when we're done here. So as I said, this is a paper that was published in iScience back in April. Adenocarcinomas are cancers of epithelial layers. It occurs in a number of tissues, occurs in the lung and the stomach, the gut, the GI tract, the bladder, number of other places. This particular paper is about lung adenocarcinomas. It's known that down here on the bottom, there's different types of carcinomas that occur in the lung. Expansion of the epithelial layer, expansion of the epithelium, making the protrusion filled with mucus. Same expansion with filling by matrix and parenchymal cells, filling with just fluid and solid tumors. So it's explained in a little bit more detail here. In our model, we're going to start with a layer of epithelial cells with some matrix and parenchyma underneath our epithelial layer. And then we're going to have our epithelial layer consisting of normal epithelial and cancer epithelial, which is what these purple and blue. So purple is cancer and blue is normal. As the model progresses, it can generate these different morphologies. This is called papillary, adenocarcinoma, micropapillary, Acinar, where it's mucus-filled, solid, where it's a solid tumor mass, and lapidic, where the tumor cells just keep proliferating but maintain the normal, more or less normal layout within the tissue. Some conclusions from the paper were that cell-cell contact energies change the tissue architecture of different LUAD phenotypes. Difference in growth rates do not correlate with the LUAD tissue architecture. Differences in adhesion, gene expression does cause changes in the tissue architecture. And modification of cell-cell contact energy change the spheroid morphogenesis. That was a follow-up experiment that was done with tumor cells in culture in an in vitro model. So the Computational model detail, it consists of stromal cells, which are these orange cells here. They have the capability to grow or shrink and or be deleted. There's extracellular matrix, which is the gray material in between, which can also grow, expand and contract and be deleted or be created. I should add creation. Well, that's what grow is. Then there's normal lung epithelial cells, like this cell right here. These are compartmental cells that have an apical compartment, basal compartment, 2 lateral compartments, and a cytosol. And then we have our lung. It's the typo. This should be the cancer cell, carcinoma cell, which has the same layout as the normal epithelial cell. It's also an epithelial, 4 compartments. The cells have internal adhesions besides the regular old external adhesions. In addition, the cells have volume constraints, but no surface constraint. Cell adhesion is just adjusted by adjusting the contact energies between the various cell compartments. In addition, as shown down here in the zoom, the compartmental cells have internal Focal-Point-Plasticity Links, which are basically connecting the centers of the subcellular compartments with springs to keep them oriented right. So there's a spring between the apical and basal that's relatively long, between the two laterals, which is relatively long, and then from the apical, lateral, and basal to the center of the cytosol. And that's done both in the normal cell and in the tumor cells. These focal point plasticities help to keep the cell, give the polar cell the correct relative orientations of its subcompartments. So keep the apical away from the basal and keep the two laterals apart. The model uses the standard mitosis steppable for doing cell division, but for cluster cells, you have to use a separate mitosis steppable that's specific for clusters so that it knows how to divide. When this compartmental cell divides, it knows how to make two new compartmental cells and attempts to get them to have the same layout as the parent cell and the same directionality as the parent cell. So in the model in the paper, what we started with was just this initial condition of some normal cells, some tumor cells in the epithelial layer, and then studied how changes in the parameters in the model result in different phenotypes after the cell has progressed, after the simulation has progressed and the tumor has developed. So you can increase the rate of cell growth for the tumor cells a little bit, a lot. You can remove the polarity on the tumor epithelial cells. You get a different morphology. You can reduce the adhesion between the tumor and the underlying normal tissue. You can reduce the polarity of the cell, that is, to tell it to lose the ability to tell the difference between apical and basal. And then finally, it's possible for epithelial tumor cells to secrete mucus in the wrong direction. Normally, these cells would be secreting out into the lumen, but in some LUAD phenotypes, mucus is secreted. Loss of polarity of the cell makes them secrete mucus into the tissue. So in the model, we explored various combinations of adhesions, polarity, secretion, growth rates, and we're able to reproduce most of the phenotypes of most of the LUAD phenotypes that are shown here. And I'm going to skip this one, just another phenotype. So this movie was part of there it goes. Was part of the supplementary material for the paper, just showing some various variants of the model depending on changes in proliferation, changes in polarity, and so forth. And so there's a little header. So this is growing faster than it should. You see the cells divide. They start to lose polarity just because they can't get out of each other's way. Reduce the adhesion to the matrix a little bit, which when the cells divide, they don't recover adhesion to the matrix. So the cells that start in contact with the matrix tend to stay there. You go very fast growth rate, the cells just lose the ability to sort themselves out and get ordered into an epithelial layer. Here's a reduction of polarity of the cells. You see a lot of the cells no longer have contact with the underlying tissue or lose the polarity with the luminal space. I can't control when I'm in view mode here or presenter mode in PowerPoint. I can't control this animation. So this one has added mucus secretion, so you get this bubble of mucus below the tumor, which tends to force the tumor up and out. So those are some of the things that were published in the paper. What we're going to do is play around a little bit with the model. Yeah, some of the simulations shown are run for different time lengths. Generally, it's run until the system seems to have decided on what it's going to do. So let's see, if you have the model and you want to try to start it, I can give you a minute. So hopefully now you can see my cursor. And I have Twedit open and the command window behind it. So this is the LUAD demo. One of the things I wanted to demonstrate was you can add other files into the workspace. And so I have added another file called notes, and it's a Python formatted file, and all it has in is just comments. You can add a file by just coming over here into the project file listing and say, add a resource, and give it a name, and give it a type, file type, which you don't have a lot of options, but just a generic Python file. At least you can use Python comments. You can also do parameters and stuff in the file. And unfortunately, this window has a little bit of a problem. It doesn't show the scroll bar it should have, so you have to expand it to find finish. So now I have a new file called test that I can put stuff in, like lots of comments. And this file will be saved with the project when I exit, when I do a save, and it'll show up again next time I run it. So I'm going to get rid of this, remove resources. Okay. So the XML, defining our cells, it takes 4 cell types to define one of our compartmental cells. So I have a tumor apical, tumor lateral, tumor basal, tumor cytosol, and then the same thing for the regular epi cells, epithelial cells, epiapical, epilateral, epibasal, epicytosol. And then we have stromal and matrix cell. The volume constraint tracks center of mass and neighbor tracker. We're not using surface. It slows down simulation quite a bit. We have contacts, pretty much straightforward, normal contacts. However, with this many cell types, we've got 11 cell types, including medium. So there's 11 squared divided by 2 plus 11 over 2 total adhesions. So there's lots and lots of adhesions. And for clarity, I like to break up the adhesion so that every time this first tag changes, put in a line break. So here's all the medium to something else, tumor apical to something else, etc. And see the lists get shorter and shorter since you only specify adhesions one way. You don't specify them both ways. Now, since we have compartmental cells, we have another type of contact. We have contact internal. And it potentially could be a list just as long as the previous list. But in internal contact energies, you only have to list the contacts that actually exist within your compartmental cells. So my tumor compartmental cells only has to have internal contact energies to the other tumor compartmental cells. And then my epiapical, etc. cells only have to have internal adhesions to all the other compartments that they actually encounter. When Twedit will set this up for you if you tell it to use contact internal, and it'll enumerate every possible configuration, every possible pairwise, so you kind of have to go through the list and edit out all the ones you don't need. So that defines our contacts, both internal and regular. We add Focal-Point-Plasticity Links. Check something here. So we have tumor apical to tumor lateral, tumor apical to tumor basal, tumor apical to tumor cytosol, etc. But they're all set here in the XML. So this is the regular focal point plasticity. And then there's another section for the internal focal point. So these internal parameters are within the same compartment cell. And it's the same thing. You have to go through all the pairwise combinations. And then finally, we have a uniform initializer, which creates the bottom half of the cell layout. And the epi cells are actually created in the Python, but this creates the bottom stromal and matrix, with creating mostly matrix cells and a smaller proportion of stroma. The registration for steppables, and then the steppable have a internally defined function that makes a compartmental cell that takes parameters about the size, the width and height, and various components of the compartmental cell. creates all those cells, and that's a function, and then that function is called in the start to create individual cells. So this function creates one compartmental epithelial cell. And in the start of the steppable, it just goes through and replicates those cells, making both the normal and the tumor cells, setting target volumes and things. So the setup, the start, finishes the layout that the uniform initializer had started, adds the epithelial layer on top. In the step function, we have the tumor growing, so the 4 different parts of a tumor cell, the lateral, apical, basal, and cytosol that grow. In addition, there's an option to make the matrix and the stromal cells grow, and they grow at the same rate, the same growth rate, times some multiplier. And changing this number changes how fast the cells grow. This is kind of a speed limit on the simulation. If the growth rate is too fast, things will just fall apart. 0.03 is still a little bit on the fast side, but if you grow any slower, it just takes too long for something to happen. So I have set kind of fast. And then we have another class for the mitosis steppable. This does the division of the stroma and stromal and matrix cells. This one here, this one here. And then there's a separate steppable to do the mitosis, the cluster mitosis, which are very similar, but mitosis steppable is needed for clusters to handle the difference in dividing a mitosis cell or a cluster cell. So that's all the bits and pieces. It's relatively long just because there are so many cell compartments and cell types that it's just a lot of bookkeeping. So this is 240 lines long, and the XML is 195 lines long. So that's the whole model. Let's see if it works. So here we have our normal epithelial cells. We have one cancer cell, and we have the matrix and the other from the stromal cells in the matrix down here. The matrix is set to be pretty diffuse, so that's these little clusters. It has a relatively small target volume, so the matrix is kind of distributed out through the stromal. And we'll let it go for a minute. It takes 2,000 steps or so before the one tumor cell divides. There it goes. It just divided. I'm not sure if it's running slow because I've got so many things going on here, or if it's just going to run slow. See what's going on here. Yeah, it seems normal. Gonna wait, let it divide again. So hopefully you've got the model, or if you're interested, you've got the model, you've got it open, and you've got it running as well. One thing I can do to make it a little bit faster, not save images. I'll cut down the pause. So you see the 2 tumor cells are getting bigger slowly. The simulation is set to run 20,000 MCS, if I remember correctly, which is about what it takes to get it to about 10,000. A little hint, if you're writing models, always end this number with a 1. That ensures that if you have steppables that do something needed, like calculations or determinations of how the simulation went, and you're doing those steppables every 10 Monte Carlo step, or you're saving screenshots every 100 multicolor steps. If you add one, if you don't have this one here, it's possible for those last runs to get skipped because the simulation stops before the steppables are all run at the last multicolor step. So if you just add an extra one onto that, you know, make sure you actually are stopping and getting all your final output that you're expecting. So now we're up to 4 cells. Growth is exponential since every cell is growing on its own. So if 4 cells grow a lot faster than 1 cell does, at least in terms of the total number of, or total volume of tumor cell, individual cells grow at the same rate. So what it's doing is it's squishing the normal epithelia, and it is starting to expand upwards. It's the only place the cells can go. And then it went to another cell division. And most of the cells here maintain basal contact. This one cell here is a serious problem. It's separated from its basement membrane. And the FVP will try to pull it back together if it can, but it may not be able to. Now we're seeing other cells having trouble as well. There just isn't enough room in the epithelial layer for this addition of tumor cells, which is one of the phenotypes, one of the LUAD phenotypes. You just start to build up a mass. So now starting to lose the apical differentiation and it can no longer keep track or keep contact with the medium up here, the luminal volume. Yeah, there's no periodic boundaries in this model. Okay, so that's kind of the base model. It consists of the tumor cells growing and dividing, and a bunch of them just divided again. And you can see it's really made a mess out of the tissue structure, forming this lump here. So as I said, this initial setup, the growth rate is controlled by this one line, or the growth rate is set by this one line in the steppable. And this is 0.03 pixels per Monte Carlo step. So I thought we would try a few different things. So this one that's running right now is the base model. The tumor grows, but the stroma and the matrix do not change. And we get this lump that looks sort of like that. And now we'll try a few other samples. So in this case, we're going to go tumor grows, the stroma and the matrix adjacent to the tumor grow as well. Before I go on, I think, so it's making a movie. That's what all this output here is. It created a movie of that simulation. I don't know if anybody's mentioned it before, but if you scroll up in your command window, you get a performance report. And the performance report tells you total runtime, that's CPU time. It's a little bit different between Windows, Linux, and Mac. On Windows, it doesn't take into account using multiple processors. On Linux, it does. So this runtime may or may not, this runtime actually could be longer than wall clock time if it's using multiple CPUs. The key thing is it gives you the time for each of your steppables. I have 3 steppables registered. And you see mitosis and mitosis steppable for clusters are using way less than 1% of all the CPU. So those steppables have almost no cost to run. This one here, volume parameter steppables, though, is taking up 35% of my CPU time. You usually don't want any steppable to be taking up that much time. There's probably a way to make that steppable to be faster, so it's not slowing you down so much. Of course, even if you've got this 35% all the way down to 1%, you'd only increase the speed of the simulation by 30% or so. So it wouldn't be a huge increase, but it would be one place that you might consider working on this steppable to make it a little bit more efficient. In any case, so what we're going to do is now we're going to turn on growth of the stroma and matrix down here below it. So as the tumor grows, or the tumor cells grow, this matrix down below it will grow as well. So that's controlled. So here's my growth step, basically the code for my growth steppable. Here's the growth rate. And these two lines, we're telling the various tumor compartments to grow by growth rate. And then do the same thing for the stromal and matrix cells. If they are in contact with the tumor cells, which is what this is checking, then stroma and matrix grow. And their growth rate is the same as the regular growth rate times some constant. So I've got it set at 0, so the previous simulation, the stromal was not growing at all. So if we give it a 1, now the stromal in contact with the tumor will grow at the same rate as the tumor, the stroma and the matrix cell. So we just turned on growth of this down here. Make sure I saved it because it takes a while to run. I'll start up again. So this will take a few minutes. This is the regular cell mitosis, and you do tell that, I do tell that, divide by the minor axis, which is what you should pretty much always do. Instead of dividing on a random axis, divide on the minor axis. As a cell grows, and if there's anything that makes it elongate, the cleavage axis would be across the short axis of the cell, perpendicular to the long axis. I actually am controlling the division of the cluster cells with this line here. Basically, it's telling it to divide on a vertical line. So I'm giving it an orientation vector. And this really probably should be calculated based on the apical basal axis of the cells. This one assumes that the epithelial cell is still aligned the way it was originally, with a horizontal epithelial layer and a vertical apical basal axis. So you could calculate that apical basal axis and do the divisions out.
Jim, how do you, so this looks like the paper, the publication looked primarily that it was a physical model over time showing tissue deformation.
Yep.
But there was some mention of differential gene expression, looking at altered genes involved in cell adhesion. I know I've got like nanostring of clinically validated breast cancer subtypes. And I know there's a guy at UCSF that's working on subtyping another, like a liver adenoma. How would you combine both types of information to gain insights on what could be driving these different, like let's say you've got the differential gene expression, and then you've got dynamical models. How would you combine the two?
The tricky, let's see, the best way to put this. The tricky part with using differential expression is that the majority of genes have only poorly defined function. So, you look at a particular tumor type and the differential gene list is 500 genes long. And there's a great challenge in figuring out, okay, what do I do with a list of 500 genes? You can certainly say,
in the case of the subtyping, like our breast cancer subtyping is a set of 50 genes across 4 subtypes. That's not that many.
Well, that's not that
It's
bad.
clinically validated.
I've never seen one quite that small. Usually when I see differential gene expressions, it runs into the hundreds. And it is problematic in interpreting that data. So what's commonly done...
clinically validated, though? Clinically validated, I mean, just random, research grade differential gene expression experiments I can see would be hundreds.
And
lists of hundreds, but...
And how many of those, even if it's 50 genes, how many of those 50 genes have interpretable functions?
I would guess with our breast cancer subtyping, the functions are pretty much known. They're detailed in the panel, but I don't know how widespread that is and how frequent that's done.
The way it's, one common approach is people will, do a gene ontology enhancement So, what are the commonalities across this set of genes to try and figure out what processes might be involved? If you can look at the list and you clearly identify something like an adhesion molecule, then you can use that directly. And you wouldn't necessarily insist on modeling a particular adhesion molecule. But if there is some change in an epithelial cell's adhesion molecules, you can say, well, the adhesion molecule went up or went down. So we can implement that in the model because it changes adhesion between adjacent cells. If you get, it's likely that a lot of your differential expressions will be genes related to cell proliferation, which means that you have to have faster rate of cell growth in your model. But you knew that from the start, right? I mean, the basic definition of tumor growth is that the cells are growing faster than they should. You also have some problem in apoptosis and necrosis. The cells are not killing themselves like they should.
So I'd say alone, how does the physical models that you're showing here help inform treatment? Don't you have to have targets to go after, genetic targets, protein targets, to make an impact?
Well, yeah, absolutely. But, that's called biology. That often the underlying problem with the underlying cause of a disease is not obviously targetable. So if a tumor cell loses polarity, now you have to figure out how you're going to target that with a drug. How do you target loss of polarity in a cell? What drugs might do that? Well, the fact is there really aren't very many drugs that do that. But nonetheless, it also tells you, though, that things are different. How is it losing polarity? Is it completely losing polarity? If you completely lose adhesion, you're talking about no longer a solid tumor, but a metastatic tumor. So it's problematic to figure out how exactly you're going to target. The other challenge with any gene expression study is that changes in gene expression are not necessarily indicative of changes in protein expression. And a given change in a gene expression is not necessarily the same across different genes.
You'd likely want to orthogonally validate with a protein panel. Yeah.
Well, that's a problem. What if your change in gene expression is the estrogen receptor?
Sorry, say it again.
If the change in expression is in the estrogen receptor, a growing cell has to make a ton of collagen and matrix proteins and all kinds of stuff in order to grow. It's got to make a whole bunch of stuff to grow. But a growing cell only has to make 4 or 5 estrogen receptor molecules to be dysregulated in estrogen. And at that low copy number, the changes in messenger might be 0. It's very low copy number genes. So the issue is, it's easy to see something that changes where the cell has to do it a lot. It's much more difficult to see changes when the cell only has to change copy numbers by 2 or 3 to get a different response.
We see that with like cytokines, right? You see a very small change.
Well, like I'm saying, I mean, estrogen
big immune response. Yeah.
Like I say, you only need a typical cell, estrogen responsive cell might have a couple of thousand estrogen receptors. But to get 2,000 of them, you might not even have to change message level at all. You just have to change rate of degradation of message, or you have to change. Actually, you would see changes in degradation of message. What you wouldn't see is changes in recycling of the receptor itself. How long does the receptor live in the cell? So it is always a challenge to go from the gene expression to a model to something that would help you. What's a druggable candidate? However, one thing that mechanistic modeling gives you is that if we go back to my early slide where it had the 5 different clinically observed topologies of the tumors, looking at a histology section or a tissue section shows those phenotypes quite clearly. Now the question is, what causes those changes? Is it just cell growth? Is it defective cell death? Is it changes in contact? Is it changes in polarity of the cells? Is it changes in direction of mucus secretion? Is it changes in necrosis of the underlying tissue. There's a lot of possible mechanisms that could generate those. So the mechanistic model tries to helps you sort out some of the possibilities of what could be causing the changes. It's always going to be a tough step to a druggable target.
So how would you differentially treat the 5 different types of these cancers? Are there known treatments already? How does this model of modeling cell behavior help in treatment?
So this is what you get when the tumor cells are proliferating, and they're also causing the underlying matrix and stroma to increase. And that could be the stroma is actually proliferating, or it could be that the tumor cells are making matrix. So now at least the result is that you have a better maintenance of something that looks like an epithelial layer. It's still getting a little shaky, but the whole thing is bowed out a little bit. And now if we go back to the phenotypes, so that looks like this phenotype. Now, how do you, again, it's always a good question. So what are you going to do next? It's unclear. The fact is drug discovery is fiendishly difficult.
Well, I guess I'm asking a broader question, Jim, is how do we take this mechanistic modeling and bring it into drug discovery? How is it being done? I'm only familiar with the profiling components because that's my background. But this is a big interest of mine. I just don't know how it's being folded in for drug discovery.
Well, first of all, in gene expression studies, one of the first things people should do is you take your differential expressed genes and particularly look for the upregulated genes, and you just search for drugs that have any citation in the literature as manipulating that gene in any way. And actually, you don't even worry about whether the gene's upregulated or downregulated. You're just looking for a lead. The second way of approaching it is the steroidal and transcriptional factor approach of what set of genes in this model or what set of features in this model are changing, and what drugs are known that affect that process or those processes in other tissues. So, I mean, historically, a tumor is a tumor because the cells grow faster than they're supposed to and they don't die. So what we do is we do chemotherapy. We just try to kill every cell in the body, figuring that dividing cells are a little bit more fragile and will preferentially die. Not the most brilliant approach, but that's a mechanistic-based approach. The next one is can you identify differences in signaling in a druggable way? So estrogen-dependent breast cancers, the treatment is anti-estrogens. In these epithelial tumors, I think it's much more complex about where the defect is and what the defect is. However, if you can winnow out some of the mechanisms, you might be able to start to have an idea where you would want to attack the system. So what mechanisms are most prevalent in this versus this? And that might give you some ideas of how you might differently treat these two. Traditionally, historically, people would do in vitro tumor models and start treating it with compounds until you stumbled on something. There still is really no good mechanistic way. There's no turn the crank, design the drug, you're done. With the exception of some of the stuff done with RNA-based drugs. That's how we got a COVID vaccine in months instead of a decade. Because it's just, you redesign the mRNA and you got a new antigen that can trigger an immune response. Beyond that, drug discovery is trial and error, largely trial and error, unless you can find in your differential expressions a gene that's changing for which there is already a drug known.
I don't quite understand how this mechanistic modeling is coming into all of what you just said.
Well, again, what's the difference? Well, this LUAD phenotypes suggests that there is something, that's this one here. There's something different in the stroma as well. This is different than this one.
You can see that from an H&E slide, though.
You can. And
Yeah.
the question is the stroma simply migrating up into it, or is the stroma proliferating? And the H&E can't tell you that.
Right. Mm-hmm.
It's impossible to get a longitudinal H&E. It's impossible to get a longitudinal sample in general.
Right. Yeah.
Unless you're doing it in culture and you're doing confocal microscopy across it or something like that. So the model can help you try to sort out why are these two different? Is it proliferation? What the paper says is the stroma has to proliferate. In order to get this, the stroma has to proliferate. If the stroma doesn't proliferate, you get this. So perhaps you attach papillary with something that inhibits stromal growth or blocks the signal between the tumor layer and the underlying epithelium.
Wouldn't you need some like synthetic lethality or something? Like then you might just get the micropapillary subtype.
It could be, and then you have to go and look at which of these are more dangerous. And it's in the paper. I just don't remember how it goes, but the survival rate changes as you go across these, and I don't remember which ones were the worst. I know lipidic is a person can live for many decades with lipidic LUAD. But now I don't remember which of these other ones were more dangerous or less dangerous. They do have different clinical courses and have different levels.
And mechanistic modeling help get at why clinically certain subtypes would cause lethality sooner, or no?
It could.
From a homeostatic perspective.
Well, we're certainly out of homeostatic control. So one thing that jumped out was you have to have the matrix and the stroma have to proliferate to get this versus this. This has to come from loss of polarity, at least in the mucus pumps, because this basic cell layer should be pumping mucus up into this upper area. It's the lung, right? It's supposed to have a mucal layer over its epithelium.
Yeah.
So this is loss of polarity. Could you have inferred that, or excuse me, loss of directionality of the pumps? Could you infer that from this? Well, this drawing is basically made from this. So yes, they understand that that's mucus secretion. This one I think is a little easier to understand as loss of polarity with retaining high adhesion. And lipidic is just very, very slow proliferation of the tumor cells. Trying to think of a really good example where a computational model helps you understand, helps you predict where to intervene. So here in this case, if you have 50 genes, differentially expressed 50 genes, this tells you which of those genes are making the actual shape changes. Because again, you're going to have, changes in expression of histones. You're going to have changes in expression of all kinds of things. ER genes, all kinds of stuff are going to be changing. But the bottom line is you have to get a change in adhesions and a change in growth in order to get the cells to bow out like that. And some signal to the matrix underneath it. And if you're going to start to lose polarity, again, that's changes in adhesion molecules. So that gives you some idea in your gene expressions, which ones are actually having the direct functional effect. Adhesion molecules, things like that.
You could bias, starting from the mechanistic model, you could say, well, these cell behaviors are being disrupted. So we would anticipate certain pathways that are involved in these types of behaviors.
I have one about the usefulness of mechanistic models. So I would say that if we can build a model that starting from these histological images can actually reproduce the different type of tumor, and when we test it statistically, it performs similarly or a little bit better or slightly less than a statistical model, one of the best that we can have, then we have some confidence that this mechanistic model has some information in it. And then we can do something that we cannot do with any statistics. So regress back to the origin. So rewind the model, and see what went wrong. And so probably try to interfere with that. Or we can even try to apply different type of therapy and do some prediction whether this particular case might or might not respond better to therapy according to the assumption of the model. The good
Like
thing
digital
about
twin?
Sorry?
Digital twin, you mean?
No, I'm talking about just mechanistic model here. So if you have a mechanistic model that you tune according to the histology images and genetic profiling something, and your model is able to capture these parameters, it puts in its mechanics and it gives you out an output. And we find out that that output is as good as statistic at giving us a prediction about the outcome of the patient, then we have some confidence that this model tell us something about this particular tumor, and we can go back and forth that we cannot do with any statistics. Statistics tells us something in one precise point. It says, okay, this is the probability that this event will occur. Instead, the mechanistic model always allow to rewind or fast forward. And so while we do this model, we can update and make it then better with the discovery. And then I believe that it will become more predictive than standard statistics. I don't know if I make myself clear. It's a little bit
I'm thinking of like prognostic diagnostics that are based on gene expression signatures. So you're saying if you take a prognostic gene signature, like a survival rate, you could pair it with a model that's also able to determine, if it moves in this direction over a certain time course, that will lead to death of the patient. And you're saying if you can align them both, or is that what you're trying to get at?
Or that you might say, okay, I have data for retrospective cases that tells me that I find out that they had a little bit less. Try to put more lymphocytes, but the next have some idea that we have tried in silico. To tune this therapy. And this is something we can try. We can do more. I'm not saying that we can do. I don't think it's a replacement of statistic or these other profiling techniques. It can be useful for both discovery, where you try to dig and inform experiments and new clinical trials, but also you can, again, when you have a mechanistic model that on which you have some confidence that you build with statistic, after all, you can rewind and go fast forward. And this is something you can do only with mechanistic models. Is a unique feat, let's say. That's my take on this.
Yeah, I mean, one of the advantages of a mechanism-based model is, first of all, it implements what you think you know in a mechanistic way, which in general omics does not do. They're just not concerned with how things happen. It just, in 30 years of omics research, 40 years of omics research, we still don't know what most genes do. We think it's an adhesion molecule because it's homologous to some other adhesion molecule. We think it's a nuclear receptor because it's homologous to another nuclear receptor. But the fact is, biology is much more complicated than that. And the mechanistic model says, this is what we think is happening. This is what we know. This is what we think is happening. Can this reproduce what we see? And if it can, that suggests what our knowledge is adequate. If it cannot, then our knowledge is inadequate. If it can, it might be right for the wrong reason. And that's problematic. In differential gene expression, the challenge is you don't know what is defect and what is response to defect. The cell knows what it's supposed to do. And a lot of the differential gene expression is probably due to the cell saying, I'm not doing what I'm supposed to do, and I'm supposed to try to fix that. But its fix is not working. That's why so many of the early attempts at, okay, here's a strongly differentiated or differentially expressed gene. It's specific for this particular disease. Let's make an inhibitor of it. And they found when they made the inhibitor, it either did nothing at all or it made the matter worse. Because actually they were inhibiting the normal response to the cell's perception that something is not quite right. And that the cell was trying to fix itself. It just couldn't quite do it. And that made a big differential gene expression. And you attack that differential gene expression, you just made the system worse. So it's always problematic using differential gene expression because you don't know if it's an abnormal response or it's a normal response to an abnormal situation. A mechanistic model, as I said, this is what we think is happening. Can it reproduce what's going on? If it can, then okay, let's build on that. If this first the papillary is caused by matrix expansion or changes in basal membrane adhesion by the epithelium, well, let's do a tumor cell model and reduce expression of that adhesion and see if the tumor cell grows the same way to validate the mechanistic model. Of course, you can do that in culture. It's that's not all that difficult to do in a spheroid model or something like that. It's not necessarily any closer to having a drug, though. But drugs are magic. Discovery of drugs has always been magic. I mean, you look at, I'm trying to think of something. The only reason there is transplant doctors in the world is because of a drug called cyclosporin A. If cyclosporin A didn't exist, there would be no transplantation in the world. Cyclosporin A is a natural product that is completely undesignable. It's got 11 amino acids in a cycle. No scientist is ever going to design that molecule. It works by a mechanism that was unknown when the drug was discovered. So actually, immunosuppression is almost totally understood because of the drug cyclosporin A. So until the drug came along, nobody knew what the mechanism of immune response was, basically, or at least how to shut it off when it's upregulated. The same thing happens in every other disease. More often than not, you understand the disease because you have a drug. More often than not, the drug was discovered by accident. Now, the mRNA drugs changes that paradigm. And people have been working on stuff like that for 30 years, antisense DNA, going back to all kinds of things. You want to inhibit gene expression? Well, I can design a molecule in 15 seconds that'll inhibit any gene you want. The problem is it won't get into the body and it won't get into a cell, right? All I got to do is make an antisense to the RNA and I've got an inhibitor, but it won't get in and it won't do anything. But that's what the COVID and the other more modern drugs, mRNA-based drugs do, is it's a delivery mechanism for DNA and RNA-based drugs. But in any case, it's a step forward. It's another way of looking at your data. It's another way of testing things. It's another way of codifying your data or codifying your knowledge of the system. If anybody knew how to discover a drug, they'd be a billionaire.
I'm curious where across the drug discovery and development continuum modeling exists. I think Lorenzo was saying, yeah, more in the drug development space as well.
Well, modeling is used in other ways. How does a pharmaceutical chemist fine-tune a drug? Can you design out toxicity, design in bioavailability, design out metabolism, design in metabolism? Sometimes you want it to be metabolized because there's reasons for wanting a drug to be metabolized. So modeling plays a role in all of that kind of stuff, but more at the molecular level for the drug itself. Modeling is used, PBPK models are still used to do first dose in human. What is the first dose in a human? What's the time course? What do you expect the blood levels to be? Where do you expect it in the urine, the feces, in the exhaled breath? So modeling in those domains are still used a lot. And they're not necessarily terribly mechanism-based, but they certainly have a mechanism underpinning. We did that one. It basically does something like that. So it's just the tumor grows, and it's growing too fast for the rest of the tissue to actually respond to it, but nothing else is going on. We can make the matrix and the stroma grow as well, and that gives you something like this, which is one of the phenotypes. The next one is what happens if the stroma and matrix next to the tumor shrinks instead of growing. So we had stroma and matrix growing at the same growth rate as the tumor was. We can also make it grow negatively. So instead of the stroma and matrix getting bigger, the stroma and matrix get smaller. We'll let that run for a while. There was a question, also a comment about adhesivity and changes in membranes. Yes, we could certainly model that, increase the adhesion between the tumor cell and the stroma or the matrix, and have the tumor grow into the tissue instead of growing above the tissue. Anytime you do something like that, you do have to worry, and as a practical modeling matter, you have to worry about if the tumor is going to go into the tissue, where is the tissue that was there going to go to? You have to have some place for it to expand out into. In this model, it might just push the sides up. But we haven't tried that. We didn't try increasing adhesion between tumor and stroma or matrix. So what I changed in, like I said, so here's the loop that does growth for stroma and matrix. The growth rate is the same growth rate that was used for the tumor. But now instead of that growth rate being multiplied by +1, we multiply by -1. So now as the tumors grow, the tumor cells grow, the stroma and matrix will shrink. And it's running, and we're still pretty early on. We only have 2 cells. But it looks like it's going to start to dive into the tissue. So, based on the question we had a few minutes ago, changes in adhesion in the tumor cells might make them more invasive, make these cells plunge into the tissue. Alternatively, in this model, basically the epithelial layer is eating the stroma below it. And that's allowing it to migrate into the stromal domain. So those 2 conflicting mechanisms that may or may not be able to generate the same final output. So that'd be an example of, well, can the models differentiate? The 2 models differentiate between the outcomes. Maybe they can, and maybe they can't. If they can, you've learned something. If they can't, then you're basically where you started, which is okay. That's usually where you end up in science anyway. So as our cells proliferate, they are penetrating into the matrix or into the stroma, mostly because they're, for all intents and purposes, mechanically eating the stroma below it. You're still losing some cells that are losing contact with the stroma. Some epithelial cells are losing contact with the stroma. So that gives us a different tumor morphology. We're at 8,500 out of 10,000 steps, and we've reached the usefulness of the simulation because we've hit the wall down here. I made this whole model smaller just so things would run faster.
A quick clarification question, actually. So
Sure.
here you're using links to hold together the different compartments of one cell. I mean, like in the compartmental cell modeling which you're doing, you're using links.
Yep.
So wouldn't high negative contact energies play the same role in the modeling? Or is it like not a good suggestion?
The model has both. It has internal contacts defined so that, for example, the apical and basal compartment really do not want to touch each other. So it does have those. However, that's usually not quite enough to hold the model together, especially if you're making things happen fast, like we are here. You get a lot of cell growth that's happening very quickly. So the FPP, the focal point plasticity links, are just a little extra way of making sure that compartmental cell holds together with the right orientations. Apical away from basal, apical, basal, and lateral all touching the cytosol, so forth. The 2 laterals not touching each other. A lot of times in epithelial models, people will also add an FPP between adjacent epithelia. So the lateral compartment of one cell has an FPP to the lateral compartment of the next cell to stiffen up the epithelia even more. We didn't include that here. We thought that was too much.
So basically, you're saying to preserve that cell structure, not to get into some randomized orientation, this FPP links will be helpful, is what you're saying. Okay,
Yeah.
thanks.
Yeah. Okay, so that one, since we told it to delete stroma and matrix, that tumor penetrated. I think, as someone mentioned, you could probably get something very similar if you just increase the adhesion between the epithelia and the matrix and the stroma. This, though, is actually modeling deletion of matrix, deletion of stroma and degradation of matrix, which are, relatively specific processes. Okay, so that was just changing this little number here, whether it's plus or minus controls whether or not the stroma and matrix is growing in contact with the tumor or shrinking in contact with the tumor. And so that's basically what it looks like if the matrix is shrinking and the tumor starts to migrate in. These are this next one's a little trickier to go do because I have to change a lot more stuff in order to make the simulation. What we want to do is, so the tumor grows, the matrix and the stroma also grows. Got to switch that growth rate back the other way. But the tumor starts to lose polarity and adhesion. So back here, we said we want the stroma to grow. So we go back to +1 in the growth rate. And now we're going to go and start playing with all the adhesions. And there's a lot of them, right? It's all the extracellular adhesions are all listed here. I've got some notes here about the ones that change. So normally medium to the apical. So low number means the apical compartment likes to be in contact with medium. That doesn't seem right. 4 to 2. I'm going to do what my note says, even though it doesn't sound right. Medium to lateral. The medium should not be in contact with the lateral compartment. So that's why it's got a big value to keep it from happening. So we're going to say, oh, that's what, now I know why the other one was 2. Basically just going to make all of these the same. And that's how you lose, one way you lose polarity. So what we're saying is apical, lateral, and basal compartments now all like medium. They no longer, the ones that are not supposed to contact medium no longer have the high contact energies. And then we're going to go apical, tumor apical to epiapical. So this is the junction between the normal epithelium and the tumor epithelium. So we're going to get rid of that low contact energy and make that disfavored. Tumor lateral to tumor lateral. We're going to do the same thing. And say it's no longer a preferred configuration. Tumor lateral to epilateral, which is the normal lateral. We're also going to make that big, so we remove that bias. 5, 1 to 40. Basal to basal, again, we're making all of the compartments with all of the epithelial contacts be the same. It doesn't matter if it's a basal to a lateral or a lateral to an apical or whatever. And then tumor basal to epibasal, make it look like everybody else. And tumor basal to stroma and matrix, remove that bias. So we've removed any reason for the tumor cell to actually want to stick to pretty much anything except for we left in either stroma or matrix to the basal compartment. Looks like I missed one of the adhesions. So it's just barely anchored and it's really not maintaining a stroma anymore. Excuse me, not maintaining an epithelial layer anymore. So we'll let that chew for a while. And they're just divided. And it doesn't even want to stick to its neighbor. We'll let that go. Now, as I said, this is not the greatest way of, this is kind of a mismatch of editing code directly And if I want to make this model go back to where it was, I have to remember all these edits I did and go back and undo all these edits. I think on Saturday, we'll talk about better ways of wrangling your parameters in your model. It's easy enough to control things like growth rate in a more logical way. Instead of being buried down here in line 159 of my code, it really would be much better to go up here at the top and say growth rate equals whatever it is now. And take this so it's not buried in the steppable. Put it up here. Now, every steppable can reference a global parameter called growth rate. Done this way, nobody can change it. None of the steppables can change the value of growth rate, though, because it's a global variable. There is a way to let a steppable change the growth rate, but you wouldn't want it to. But you put it up here. If you want to change, now what you can do, have and I'm just using these as dummies. So I might have, so this is my tumor growth rate. This might be my parenchymal growth rate. What's happening over here? Oh, they're just drifting away. So I could have a stromal and some other parameter. Now, by changing this one variable, this be x, I just select between my parameter sets. And I go through and I hunt down all my parameters I've got scattered throughout my code and pull them all up into this one place. You can also access things in the XML. And I'm not sure if I'll be able to do this on the fly. J M E A. So this is the J value for medium to tumor apical. Once I put this tag in there like that, I can access this entire slice of the XML in Python. I'm not going to remember how to do it. And I don't think there's a helper for it. Okay, at some point in your XML, any section you want, you add ID and then some unique character string that helps you figure out what it is when they're referring to it. And then that's the code. So the element I want to fetch is this guy here. Might as well use the same name everywhere. So this says, go into the XML and get the thing that was tagged as JNPA, store that in a variable. We'll just use the same name again. Then take the C data. I'll explain in a moment. Well, no, I got ahead of myself. So take the C data, make it a floating point number, add 10 to it. You don't really need the decimal point. But in another experiment set of parameters, maybe we subtract 10 from it. We do something else to the value, or completely replace the value. So we could also do. No, leave it alone. So what is C data? In the XML, this is always the C data, the stuff, numbers between the XML tags. This is always called C data. In some commands, for instance, in this command, there's a parameter called neighbor order. So if I wanted to change this, this is the C data, but there's also a thing called neighbor order. So in the Python, it would instead of this, it would be, well, this would still be correct because I'd use the same tag, but this would change neighbor order. But this JMTA tag is an entire XML line. And that entire XML line already has this hard-coded thing called neighbor order. We'll go back to put it back to what it was. So this is a way of, okay, I've got experiment 1, and these are all the parameters I changed. And I don't have to hunt down my values, and I don't have to keep going back and forth, and don't get confused about what I'm doing. You can also do handy things like, say, parameters can be functions of other parameters in your model. This is a great way of doing it. Now, if you want to get even fancier, what you do is you would actually put this in another file. A resource. We'll call it parameters. I spell it right? Almost. And it's a Python. And our missing button. We go over here into parameters and go. Might do something like now. 1 pixels equal 1 micron. 1 MCS equals 1 minute. Just so you got a note, it's written down somewhere what you thought your units were. Now all my parameters are in this file. They're all gathered in a nice, neat, tidy place. I use it in my Python. I'm just comment this out. I typoed a whole bunch of stuff there. So this parameters is just a Python file, and you can import a Python file. So I'm going to import my parameters file, but I'm just going to call it P because I can't type parameters without misspelling it 3 out of 4 times. One thing that's just weird about this, that if you do this, it'll crash. I do not know why import does not allow a file extension. The file is actually called parameters.py. Here we can go. So to refer to one of the variable names in our parameters file, we imported it as P, and you just stick P dot in front of it. So now instead of having my variables scattered all over the place, this is a little bit better solution. This is an even better solution because now I can also do stuff here like define things in terms of microns and pixels, or excuse me, microns and minutes, and then do the conversions. And I can even do stuff, more complicated things like and then in the Python, change the dimensions of my comp cell model using this value in here. So there's a lot of things that you can do this way, defining your parameters, trying to assign units to your parameters so that you actually, at some point in time thought that your diffusion constant, which was quoted in the literature as centimeter seconds or something, that works out to be such and such pixels per MCS. Lets you define different parameter sets and pretty easily switch between them. So now we're going to do experiment 2. So that's a real handy way of trying to keep things a little bit more organized. Let's see if that simulation. So we just made a metastatic tumor. It's metastasized and it's going to go everywhere. A little quick visit of epithelial layers and uncontrolled growth and changes in adhesion, making it do slightly different, making it do different things that look like some of the clinical phenotypes. And a little bit of a discussion about how you might want to wrangle parameters when you're trying to get the model to have more than one version of the model that behaves differently, and ways of keeping track of your parameters and variables and so forth. So there's another version of it if you remove the polarities. So there's a couple more examples, and then might want to think about it if you have time. This particular example, the tumor grows, the stroma grows, matrix adjacent to the tumor grows. The tumor just loses its epipolarity, but it still has its adhesions. So the tumor folds in to the underlying parenchyma. And this was a question if anybody wanted to try something different and what happens if you lose the internal polarity. So you remove the FPP links within the cell, within the compartmental cell, and then change all the adhesion so that it's perfectly fine for the basal compartment to touch the apical compartment. Some critiques of this model. The cells grow and divide nearly in sync. I think you saw that when the model runs. The cell gets bigger and bigger and it divides, and then those two get bigger and bigger. And those two divide at almost the same time. It's mostly because they have the same growth rate and they all start at the same place. So this could be fixed by changing the growth rate for each cell. Pick a growth rate for a particular cell from a distribution about some normal or standard growth rate, so that each cell grows a little bit different rates. And then perhaps do the same thing with the doubling volume, how big the cell has to be before it divides. Again, assign some distribution about a standard value so that every cell behaves a little bit differently. The compartments all grow at the same rate, even though the compartments have different sizes, different nominal size or different percentage sizes of the cell. So their growth rate really should be scaled by the size. And the thing I mentioned earlier, this particular code is spending way too much in the volume parameter steppable. I think it's most likely because it's doing something every Monte Carlo step, and it's almost never necessary to do something every Monte Carlo step. Make the growth rate 10 times faster, but then only apply it every 10th MCS, and that would probably knock this number way down. So that's it.